Okay, sorry. I thought we were good, but now we're good. Hello. So yeah, I am kinda, maybe, ish on time. Sorry, I'm about a minute or so, 2 minutes late. 3, probably. Oh well. Shit happens. I'm sorry. So like, I am in the middle of Satan's butt crack at the moment, because it's sweltering and hot. And I cannot sit in this room for like 2 hours. Normally, I stream for like at least 2 hours. Yeah, that's not gonna happen today. I'm thinking about 45 ish minutes. So, yeah. We might have lag. I am sorry if that happens. My bad, but I hope you understand that this is quite an extreme heat wave that we are going through here in Europe. And my computer needs a little bit of coolness, and everything is just running on like its last sip of energy. So it's a thing. So before we start and jump right into the major news about Homo Naledi, I want to look at the merch store, and I ordered one myself. So this is the Homo Heidelbergensis shirt that I designed, as you can see. It's beautiful. It's got the skulls on the sleeves, and you got the 3 skulls on the back. I mean, doesn't that look gorgeous? I mean, okay, yeah. I'm biased because I like skulls and not everyone likes skulls, but I mean, come on. This does look good, right? This does look good. So, yeah, get your merch at historywithkaylee.com. Yeah, and highly recommend because my merch awesome. So, yeah, okay. Now that we've done that, I can also say, join me in Turkey. It's gonna be awesome. I promise. Like, seriously, consider joining me in Turkey. So the link to that is in the chat in 3 seconds. 3 seconds. There we go. There's the link. There you go. Okay. All right. So I wanted to do a live stream about this new discovery about Homo Naledi. Homeowner lady, yes. Also, one of the t-shirts on the merch store, by the way. Where is it? Where is it? Skull Collection. It's in the Skull Collection, of course. And it is here. The white Naledi and the dark Naledi. I mean, come on, guys. You can walk around with your own Homo Naledi t-shirt. Or, you know, you could just wear Homo Naledi, homeowner lady. It's also still in the store. But yeah, grab yourself a nice frickin' tea and let's get into the news because it's sweltering hot and I need to stay awake for at least another 7-ish hours because the Netherlands plays in Kansas tonight against Tunisia in the World Cup. And of course, I will be wearing my orange t-shirt and I will be supporting my team, my country. Come on. I like the World Cup. I love football. I try to watch everything. So, yeah. Okay. Oh, so, shit. We also have, of course, the Twitch stream that I need to turn on. Because there might be people in the Twitch chat that are like, woman, why don't you understand that I'm here? Usually, no one watches me on Twitch, but that's fine. That's fine. We keep trying. Okay? We keep trying. Mm-hmm. Also, I might be getting better internet around August, so the streams might be more streamlined. Pun intended. Yeah, I'm not funny. Okay, let's jump right in. So scientists, Lee Berger and his team, they have found the Homo Naledi skeletons inside the Dinaledi chamber of the Rising Star Cave, and there was also another chamber, but it's found in the Rising Star Cave system, which is located in South Africa, which is also the broad cradle of humankind. And so they did that discovery a couple years ago. I made a video on that. Y'all really loved it. I did a couple follow-up videos on that. They did okay. I interviewed Dr. Lee Berger twice on the channel, so that's 2 separate videos. I think most people really enjoyed that. There's a lot of people that are contesting the discoveries, and so they continue doing their research, as they should. Let's be honest, as they should, which is really good. And now they did genetic analysis, like a protein analysis of the Homo naledi skeletons that have been discovered. And there's a curious twist to all of this. None of them seem to have Y chromosomes. So that means that all of them were female. And that begs the question, where are the Homo naledi males? So we're doing this live stream today. I'm working on a recreation of my ancient air conditioning video that I made years ago. That's going to be the next video because heatwave was a good reason for an updated version on that. And then next week or later this week, probably early next week, I will make a video, a dedicated video on this discovery itself as well. But I thought, you know, why not cover this in a video on the live stream first? So like people that watch the live stream will get the information, and then if people don't watch the live stream but still want the information, I can make a dedicated video on this same topic as well. So, you know, let's do that. So here we go. There's 2 articles that I grabbed, and this is the dedicated publication Naledi, and I'll put that in the chat as well. So there you have it for the people that like to read scientific publications. Not too many people do, I understand. That's why we're going through the articles about this topic and not the scientific publication, because there's a lot of big words and a lot of stuff, and not everything is as interesting as it may seem. It's very dry to read that kind of stuff. Only weird people like me enjoy it. So if you enjoy it, see it as a compliment that I see you as the same sort of weird people that I am. So, I mean, it's all good. It's all good. It's all good. But I did want to share it with you guys to give you at least like the option of looking into it a bit more yourself. So, around 300,000 years ago, a cave in South Africa became the final resting place of at least 20 human cousins from the ancient species of Homo naledi. Today, researchers have determined that all of those individuals appear to be missing a Y chromosome, the telltale genetic marker for males, and they are likely female. So these are not necessarily, like, these aren't skeletons of like infants. So they would have had a Y chromosome if they were male. Okay? Okay. Now that we got that, let's go. So using a new method of sampling ancient teeth, the team was able to collect chemical clues about the sex of each skeleton without destroying it. The technique has the potential to transform paleontology for several decades, says Lee Berger, who is the National Geographic Society's explorer in residence, paleoanthropologist at Wits University in South Africa, and a co-author on the new study. Berger and his colleagues first discovered Homo naledi's bones in the cave system called Rising Star in 2013. Following a tip from 2 cavers, the team uncovered around 1,500 fossils in a chamber deep underground, only reachable through a narrow chute at the end of a twisting 100-foot passage. At the time, the team assumed that some of the larger skeletons were male, and that the species had relatively equal numbers of males and females. But the common pattern, like the common pattern in modern mammals, finding a massive trove of ancient bones from only one sex is almost unheard of in paleoanthropology. But the samples from Rising Star Cave that had previously been identified as male now appear to be female, researchers reported on June 24th in the journal Cell. So that's the link that I shared with you guys. For the people on TikTok, I can also share that with you. I'm not sure if it's like clickable on TikTok, but I can at least send it. And if you want to look into it yourself, go. Or you can just, Google where are like Homo Naledi female, and you will probably find the article or like the publication real fast. So here you can see like the Rising Star cave system. It's located around 25 miles outside of Johannesburg in South Africa. So these are very exciting results, pointing to the cultural and perhaps even symbolic significance of the Rising Star site, says Katarina Duka, an archaeological scientist at the University of Vienna in Austria, who was not involved in the study. Barring alternative biological explanations, she sees the find as a unique and perhaps perplexing prehistoric discovery. This isn't the first time that Homo naledi has challenged assumptions. The ancient cousin's strange mix of traits has long confounded paleoanthropologists. A brain that's 1/3 the size of our own, shoulders optimal for climbing like those of older human relatives, and long legs capable of strides, similar to modern humans. Berger's team has argued that at Rising Star, Homo naledi intentionally buried their dead, stoked fires, and carved symbolic cross-hatchings into the cave walls with stone tools, achievements often associated with big-brain species like us Homo sapiens. The new finds deepen the mystery around what the ancient hominins, the umbrella term for humans and our ancient relatives, were doing in that cave. And Berger thinks it strengthens the evidence for advanced abilities in Homo naledi. It should give you almost goosebumps, says Berger. You're looking at a non-human species with the brain the size of a chimpanzee that is saying females have a separate place in this world and an important place, in that this place belongs to them in death. Where? So, wow. So how to determine sex in hominin fossils? DNA is the most surefire way to determine the sex of a skeleton, but it degrades over time, especially in warmer climates. While researchers continue to push the limits of recovering ancient DNA, the current record holder comes from an environmental sample from 2.4 million years old of ice in Greenland, and the oldest DNA from a hominin is around 400,000 years old. Even when DNA survives, holes in the sequence can limit the tests researchers might run on ancient samples. When DNA is not an option, which it often is not, researchers examine the bones themselves. The shape of the pelvis and skull can vary between males and females, as can the size of teeth. These morphological features are fairly reliable, but one can rarely be 100% confident in them, says Paul Constantino, a biologist at St. Michael's College in Vermont, who was not affiliated with the new analysis. But ancient hominin fossils are often so rare that these variations are hard to discern, leading to debates over whether a skeleton represents a different sex or even an entirely different species. So here you can see two pictures. Berger's team excavated over 1,500 bones at Rising Star, some shown in this excavation pit. Based on the distribution in the cave floor surface sediment, the team suspects that the fossils were deposited there over a long period of time. And here in this ultraviolet picture, during some of the field excavations, researchers used ultraviolet light to reveal some of the bones buried in the sediment. Okay. Now that we've gone over the pictures, we can continue scrolling. So because of the limitations of bones and DNA, scientists are increasingly turning to ancient proteins stuck to teeth and bones to answer some of these big evolutionary questions. Sequences of proteins can tell researchers where a species might fit in the human family tree. Molecular biologist and study co-author Enrico Cappellini's lab at the University of Copenhagen in Denmark mentioned, has used such sequences to reveal that modern orangutans are related to a 1.9 million year old giant ape, Gigantopithecus, and shed light on where ancient rhinos and human relatives fit in their respective family trees. I also just realized I never turned on my lights. Give me a sec. Give me a sec. One sec. I'm sorry. My bad. I was not well prepared today. Not at all, actually, because it's too hot to even think. Okay. I'm back. That's a little bit better. That's a little bit more light at the end of this tunnel. Okay, sorry. I'm back. Here we are. So these protein sequences can also illuminate the ratio of males and females in samples of bones. A protein in tooth enamel called amelogenin looks slightly different depending on whether it comes from a gene on the female X chromosome or a gene on the male Y chromosome. Last year, an amelogenin analysis by the same team showed that the females of the 2 million-year-old hominin species Paranthropus robustus weren't necessarily smaller than males, bucking a common primate stereotype. Really cool. Really cool. So, a remarkably homogeneous hominin. Based on that track record, Berger, Cappellini, and their colleagues wanted to use ancient proteins to tackle a number of questions around Homo naledi. They hoped sequences might hint at where the species fits into our own lineage, as well as how much the individual sequences differed across skeletons, which can tell them how diverse the species was. First, the team sampled just 4 individuals from the cave, removing a piece of each tooth to test the enamel and see if they could sequence its proteins. But they noticed something odd. All 4 lacked the Y chromosome variant of amelogenin. So the researchers tested more teeth and even tried an alternative method that was less destructive, involving scraping the enamel off instead of cutting out a chunk of tooth. And the etched sample is no greater than what a dentist would do to roughen up your teeth to attach braces, says Berger. Okay. So here you can see Neo, the skull of Neo, one of the Homo naledi skeletons that they found. The skull nicknamed Neo represents the largest member of the Homo naledi skeletons, while a skull labeled DH3 is among the smallest. The new result suggests that both are female. So we can have big females and small females. Just like with humans, I'm a tall person. I'm 174 centimeters tall. And for my American viewers, that's 5'9". Not very small. I'm very average compared to like other Dutch people. But when I went to Peru, I towered over most people, which was weird to experience. I've kind of towered over people in Turkey and Egypt, but in Peru it was way more obvious. So out of 20 individuals, the Y chromosome was definitely missing in 19 of the samples and likely missing in 1 outlier. Even a tooth belonging to an individual dubbed Neo, whom they had previously identified as a male, was missing the Y version of amelogenin. And welcome to the Dolmen level Silvorathon. Good to see you. Yeah, it's about a human relative, actually. It's not an ape. It's a hominin. So this is a Homo species. So just like us, it's a human species, but they went extinct a while ago and they were a lot more archaic than us. But they are a human species. So yeah. So we were all shocked, says Palesa Madupe, a molecular biologist at the Max Planck Institute for Evolutionary Anthropology in Germany and lead author of the work. Only one other ancient site, a cave in Portugal, has a female majority among skeletons, which researchers have linked to the mortuary practices of local Neolithic humans. While the Homo naledi skeletons included 10 adults and 10 juveniles, including an infant and some toddlers, none of the children were male. That finding is particularly curious. Children rely on their mothers so much, one would expect to see some male babies. Even most Homo sapiens societies that separated dead individuals based on their sex keep male infants with their mothers, says Berger. Yes, we are from Africa. I've also made a video explaining the whole out of Africa theory and why it is still the prevailing theory and correct. Yeah, and a scientific theory, an actual scientific theory, isn't the same as just a theory. Like, it's been proven time and time and time and time again. That's why it's a scientific theory. Like, they tried to look at everything that could ruin that theory, like ruin the whole basis of the theory, and they couldn't find anything, which is why it's the prevailing theory. So, unlike other species such as Paranthropus robustus, Homo naledi's bones have a similar shape across individuals. For this reason, George Washington University paleoanthropologist Bernard Wood, who did not work on the new analysis, says he wasn't surprised that they might all belong to one sex. He points to a hand bone in particular. They look as if they've been made in some sort of stamping machine, he says. There's no variation at all in the morphology, which is very strange. The protein sequences of each individual were also remarkably similar. Wood suspects that this population may have been isolated in South Africa, perhaps descending from just a few individuals and thus retaining less variation in their genes. Here you can see how they climb into the chamber through this narrow chute. Like, it's really, really, really narrow. Berger actually went into the Dinaledi chamber a couple years ago when right before I interviewed him, and there were moments where he wasn't sure if he was going to be able to get out, but he was full on accepting the fact that if he did not come out, that he would die there, which is crazy. But yeah, the only way to access the important Dinaledi chamber, in which most of the site skeletons have been found, is through a narrow vertical chute. Here you can see Dirk van Rooyen, the principal field coordinator of the National Geographic Society Rising Star project, squeezes through the very tight and awkward slot. And here you can see the inside of the Dinaledi chamber. Modern humans first entered the famed Dinaledi chamber on September 13, 2013, through a narrow chute, that you can see there, discovered by recreational cavers Rick Hunter and Stephen Tucker of the Speleological Exploration Club in South Africa. There they found an astonishing array of fossils. Okay, yeah, exactly. Dinaledi chamber, exactly. So Homo Naledi was in the Dinaledi chamber. I agree. Okay. Goddamn. From biology to culture. The probability of finding all females among 20 random humans are staggeringly low. Specifically, 0.0000954%, according to the team's calculations. The question now is how this could have happened in Homo naledi's case. In very rare instances, the amelogenin gene on the Y chromosome has been known to vanish. A small fraction of males in some modern human populations have lost the Y version of the protein due to mutations. The rates are low, 0.18% in an Austrian population and 8% in a Pakistani population. There's also the possibility that the Y chromosome has degraded more than the X chromosome over time. Female false positives can happen in those cases, notes Carles Lalueza Fox, a molecular biologist at the University of Barcelona in Spain, who was not affiliated with this study. For that reason, he remains unconvinced that all of the skeletons are truly female. Only additional testing could rule out false positives. However, the authors argue that the detection of the male version of amelogenin in both Paranthropus robustus and another ancient hominin called Australopithecus afarensis, both older than Homo naledi and thus more likely to experience that degradation, that makes this unlikely. So, if the Y chromosome hasn't gone missing and the Homo naledi individuals are indeed female, then some researchers wonder if some unknown ancient hominin behavior could be at play. So there's a lot of ancient extinct human species. I think 9 that we know of. But okay. Constantino speculates that groups of Homo naledi may have had fewer males, similar to the single male social structure seen in mountain gorillas or hamadryas baboons today. Or the species may have divided labor based on sex, and perhaps the males primarily hunted or patrolled the territory. Scientists don't yet have enough clues to fully comprehend how Homo naledi might have used the depths of the Rising Star cave systems. The one certainty is that its dark chambers became their home in death. Okay. I get cat hair sticking to my nose, and it's itchy. Don't live in a heat wave. I'm just telling you. Do not live in this heat dome. Okay, so at one point, Rising Star Cave's largest entrance was used by old miners to excavate the calcite speleothems from the walls and ceilings. Martin Gamache in the background, Tegan Stoop and Dirk van Rooyen in the foreground peer out over the expansive chamber. Really cool. It's a really cool cave. Awesome. So yeah, cool. Wood, who doesn't believe that Rising Star contains intentional burials, like I said, there's a lot of people that are contesting all of the Homo naledi stuff and research and discoveries and publications. And I'm not a paleoanthropologist. I'm not an anthropologist. I'm not a paleontologist. I'm just an amateur who never finished high school. I just do my own research. I just grab what I find interesting. I research those topics to the full extent that I'm capable of, and then I make videos on it. None of my videos can be seen as 110% factual because I am an amateur, but at the same time, I have anthropologists, paleoanthropologists, paleontologists, geologists, historians, archaeologists, all of them come to me and they tell me that I do great work. So I'm kind of inclined to believe them. Sorry, I'm just saying. They enjoy my work for the fact that I do what an amateur is able to and give that information and share that with as many people as possible. And I've never had them tell me that I outright did or said something wrong. Do I personally believe that the Homo naledi burials are intentional? After speaking with Dr. Berger in that interview that I did with him and him showing me all the evidence for it, I personally believe that yes, it was intentionally buried. Like these fossils were, like these people, these humans were intentionally buried. I do believe that. Do I believe that they created art by scraping the walls? Well, I do believe that the scraping of the walls is a form of art. Do I believe that it was Homo naledi who made that? It's highly possible, yeah. Where is that hair? Please go away. Go away, it's so itchy. But I like I said, I spoke with him. I did that interview with him. He showed me all the evidence. Yes, I believe him. I personally do not contest it. But at the same time, I am just an amateur. I am not able to discern intentional burials from non-intentional burials. I'm not able to discern intentional artwork from just scrapings on the wall. I'm not able to discern that that artwork was definitely 100,000% created by Homo naledi. But the proof that Dr. Berger showed me makes me inclined to feel that he's correct. But at the same time, I'm still an amateur. So, yeah. Okay. Just wanted to preface that because, like I said in the beginning, a lot of people can contest all of the Homo naledi finds. So, I understand. So, yeah. So, again, Wood, who does not believe that Rising Star contains intentional burials, where individuals might have dug graves or arranged bodies, wonders if Homo naledi may have taken female remains to the cave, perhaps dropping them in after death, Others have suggested that the bones may have been transported by water or predators to the cave, but each theory has their own limitations. Aside from some form of cultural behavior, there is really no other explanation for why you would have just one sex in the cave system, he says, at least not a natural one, which makes me feel more inclined that they were indeed intentionally buried. Weird, right? Yeah, no, I know. Berger thinks that the exclusion of male skeletons is evidence of a practice that would have required a level of complex thinking only seen in humans. We are sitting with evidence of separation at death by sex, and thus likely gender, he says. That is clearly evidence of culture in a non-human species, even though this was a human species. So I'm not sure like what that end of the sentence was, but if that was a direct quote from him, okay. But Beatrice de Marchi, a biomolecular archaeologist at the University of Turin in Italy who did not work on the analysis, is still cautious about drawing cultural conclusions. She notes that beyond evolutionary patterns, it's incredibly difficult to interpret biological data from the past, especially with limited or unique context, as in the case with Homo naledi. In this sense, Homo naledi is a classic case in which the boundaries between nature and culture are blurred, she says. Nothing but the truth. More broadly, the study shows just how valuable proteins can be in unveiling information that remains ambiguous in bones or absent in degraded DNA. Constantino called the less destructive sampling method a breakthrough in paleoanthropology. Much of the hominin fossil records consists of teeth in sizable numbers at some sites, he says, and knowing how many individuals were male and female could point to how these ancient groups were structured and even mating trends. Multiple researchers expressed hope that the technique could help sample ancient hominin specimens from East Africa like Paranthropus boise, which has also faced questions around its legitimacy as a species. It could also validate preconceptions of sex for famous fossils like Lucy, or settle debates over the sex of enigmatic fossils like Mr. and Mrs. Ples, the most complete Australopithecus africanus skull ever found. It is wonderful to see how ancient proteins open up the window of prehistory a little wider, bringing us closer to our hominin cousins, Duca says. As for Homo naledi, the mysterious human genus still has no clear ancestors or descendants. The case of the missing Y chromosomes still plagues Madupe, who wonders if there's some explanation for the absence of the male protein signal lurking in the unknown realms of molecular biology. If so, that will be a great discovery, she says. Only a definitively male skeleton could answer that question. Berger estimates that there could be dozens more skeletons in the cave system ripe for discovery. Clearly, there had to be males, otherwise the species would not exist, Constantino says. So, where were they? For now, the new revelations have a strange serendipity to them. The tight squeezes required to enter the present-day Rising Star cave system made it harder for male researchers to enter. So the initial group of paleoanthropologists to excavate the bones were all women. That these female fossils were discovered by women, Berger notes, I think is one of those great beautiful ironies of science and exploration. So this was the National Geographic coverage about this new publication. Again, the publication is in the chat. I think it's really cool. I also found a John Hawkes article, and I very much enjoy reading a good John Hawkes article. So even though about 80 to 90% of the information that I just read will be the same, I still want to read this John Hawkes article because I very much like his writing style. I very much like his articles, and I don't know, like I'm a fan, okay? I'm sorry. I'm not fangirling, but like I am a fan, and holy shit, there's still a cat hair stuck to my nose, and it's so itchy. Everything is just stuck, like sweat, and everything is just, ugh. Heat wave! It's pain. I don't want to live in Satan's butt crack anymore, people. Please save me. Give me air conditioning. Oh my god. So, yeah. Okay, awesome. Okay. The first molecular data from Homo Naledi, protein data from the rising star cave system, shows the known sample of the species is entirely female. Why no AC? It's expensive to buy, it's expensive to run. And our home is really not well suited for it. So, I'm sorry. No AC. Most people in the Netherlands don't have AC. But also, it's normally not this hot and this sweltering. It's a heat dome. It's extreme weather. It should not be this extreme. Okay. Okay. It is great to be able to share some of our team's research on the proteome of Homo naledi, published today in Cell. The study is the largest examination of the protein data from any sample of an extinct hominin. The work has been done on teeth of Homo naledi from all parts of the cave system where we have recovered their remains, amounting to at least 22 individuals. How hot is it? It's currently 29 Celsius, but like the inside of my house is 27 Celsius. And tomorrow it's getting hotter, and this upcoming night that we are entering in a couple hours will be the hottest recorded night ever in the Netherlands. It will not cool down tonight. Not at all. And we only have like 6 hours between sunset and sunrise. So there's no time and nothing to cool us down. So, yeah, it's not awesome. I feel for the people in Spain because, or France. I'm sorry. I mean, I feel for the people in France because I know they had the brunt of this heat dome, and it's currently like slowly going northward. It's crazy. It's crazy. So, so we'll read this article, and after that, the stream is just done. But like I said, it's a shorter stream. We're already doing almost 40 minutes, so like we're doing good. But that's why I recreated my ancient air conditioning video. I just wrote the script today, like finalized it, and it's gonna be good. But yeah, if I ever have enough money to build a home from scratch, I will definitely make sure to have a wind catcher system in that. Even though it's not necessarily completely suited for the Netherlands, I will still incorporate it. Even if I have to put mechanical fans, like running on the electrical grid to help that wind go through the house, I don't mind. It will be better than nothing. So yeah. Yeah, exactly. 29 Celsius inside of your house is not okay. Because the humidity. So the humidity today in the Netherlands, let's check. Because like I said, tomorrow will be way worse, but let's check humidity in Netherlands today. 73% right now. 73% humidity. Right the F now. And tonight, like I said, it won't cool down, so the humidity also will not go down. It will probably go up. And then tomorrow, it's going to be probably the hottest recorded day in the Netherlands ever, so that's it's fine. And people that rent also can't use AC. And window units don't really work. And the portable units don't really have enough power to cool, like, a living room. Or a bedroom. Only a small room. So it's just, AC is, it's not, it's, you know, we have fans in the house, like we've got vertical, like tower fans, and we're okay. The cats are okay. I'm okay. This office that I'm in is just extremely hot because it's the sunny side. As you can see, the sun, like I have to like block the sun with a thing over the camera because otherwise you won't see anything. Doggerland, the documentary, it's still coming. I'm promising. I promise. I promise. I promise. It is still coming. I'm so sorry. I had some sponsorships that I had to do. I got sick a couple times. My body is not doing what I want, but it's fine. Yeah, it's fine. We're okay. Everything will be okay. Doggerland documentary coming. I think first or second week of July. Probably second week of July. Let's just be honest. I got ADD and I got like 10,000 things going on in my head at the same time, but it's fine. Okay, so John Hawkes, first molecular data from Homo Naledi. The most fascinating finding is the simplest. At least 20 individuals show clear evidence from protein of their chromosomal sex. None show any signs of male markers. Adults, children, infants, all the individuals have protein markers consistent with the female sex. It has been a great experience. Probably should make this a little bit bigger for the people that want to read along. I'm sorry. It has been a great experience collaborating with some of the leaders in ancient proteomics, especially our lead author, Palesa Madupe, now with the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, and Enrico Capellini, one of the leaders in ancient proteomics at the Global Institute and University of Copenhagen, again, Denmark! And we will see Henrik in the chat doing Denmark with the flag and everything in a couple seconds. It's like a 3, 2, 1 thing, and he will be there, at least if he's in the chat. There are 21 authors, all of whom were essential in different aspects of this project. I'm excited for where this collaboration is going next. Here you can see 6 of the 22 individuals represented in the study of dental remains from the Rising Star Cave system. This comparison shows individuals across the lifespan from a 2-year-old infant at the top left through young and older children, adolescent and young adult, to the oldest adult in the sample at the lower right, with teeth worn to the roots. Tooth enamel contains a fairly limited set of proteins, most related to the enamel structure. One of those proteins, amelogenin, has 2 versions that are products of 2 different genes. Amel X on the X chromosome and Amel Y on the Y chromosome. These differ slightly in amino acid sequence that makes peptide traces of the amelogenin protein useful as a marker of chromosomal sex in ancient samples. Individuals with we have Barry. Barry did it. So Henrik's probably out of the chat, but at least Barry did it. And Ron's digging in the Midwest too. Like, Denmark represent. Okay, perfect. Individuals with 2 X chromosomes have only AMEL-X peptides. Peptides that are part of AMEL-Y are only found in individuals with a Y chromosome. This method has been applied in archaeology now for a long time. In archaeological sites of the last few thousand years, testing for amelogenin can be done with a very small residue of enamel. In much more ancient remains, like extinct species of hominins, the short segments of protein that survive, called peptides, have very durable molecular bonds and tend to last a long time. Like I said, by the way, we just read the National Geographic article, which was a good article. John Hawkes, the level between writing styles and just the amount of information and the way the information is conveyed, I love John Hawkes' articles. Like, they are absolutely amazing. I love reading them. It's so clear, it's so written, like in such a good way and style, and you just roll in it. I love it. So, how long do those peptides last? How long? Previous work from Palesa, Enrico, and their laboratory has recovered protein data from teeth of Paranthropus robustus and Australopithecus africanus from South African sites, up to 3.6 million years old, including individuals with AMEL-Y in both these species. Earlier work on Homo antecessor found the AMEL-Y marker, as have many studies of Neanderthal and Denisovan individuals, and the recent work on Homo erectus teeth from China. Beyond hominins, several Miocene age sites with more than 15 million years old have yielded protein data from non-hominin faunal teeth, including in the Turkana Basin of Kenya. There is a great deal of potential for understanding the sex of hominins and in fossils of other families of animals from this approach. The Homo Naledi teeth are recent compared to some of these finds, a mere 300,000 years old. Good. One of our team's priorities has been to support research on less destructive methodologies to apply to the fossils. Because there are so many fossil teeth from the Rising Star Cave system, it is a valuable collection for validating methods that can later be applied to sites where they are more scarce. Palisa, Enrico, and other lab members have applied a number of experimental approaches to compare different methodologies. In a practical breakthrough, they were able to show that a slight acid application on a small area of enamel can generate as much peptide data as more invasive physical sampling. Even as this new paper has been in preparation, both this new approach and similar ones have been successful in finding peptide data, including male markers from many fossils, including the much older South African fossils and the recent work on 400,000-year-old Homo erectus fossil teeth from China. The Homo Naledi teeth in this study represents all ages from infant to the oldest adult in our sample. Around half are children. They come from every part of the cave system where we have recovered fossil material, including both the Dinaledi and Lesedi chambers, the Hill antechamber, and the UW110 locality where the skull and teeth of a Homo Naledi child were recovered. All are female. An obvious question with any biomolecular method is whether the laboratory methods have the power to make a strong conclusion. In this case, both the extraction methods showed the same results. Several different analytical approaches coming from multiple groups were applied to understand whether the lack of amyl Y evidence could be explained by the preservation or recovery of the data. All of these converged on the conclusion that the results are certain for 20 individuals. If these 20 were male, there should be amyl Y markers. For 2 additional individuals, no male markers were found, but the lower amount of peptide data recovery makes the identification less certain. In our paper, we mention alternative hypotheses, such as the idea that Homo Naledi had intersex karyotypes, or genetic changes to the Y chromosome that resulted in amyl Y inactivation in all male individuals. Such genetic scenarios are much less likely than the simpler hypothesis that the sample is female. As I'll discuss further below, the morphological data from the sample also supports the female sex of the sample. There are other enamel proteins besides amelogenin. Their variation has some potential for understanding the evolutionary tree. So far, there is no strong evidence from the protein data about Naledi's relationships. This is a challenge in protein data compared to DNA evidence, because there are extremely few variations that separate known populations of humans and other hominins from each other. Modern humans, Neanderthals, and Denisovans differ in frequencies of a few enamel protein variants, but small fractions of people do carry known Neanderthal and Denisovan variants. The study from earlier this year on Chinese fossil hominins by Chao Mai Fu and co-workers showed a marker shared by some teeth attributed to Homo erectus, to Denisovans, and to some living people. One amino acid change sets Homo antecessor apart from all 3 of those later groups. Last year, the study that Palesa led on South African Paranthropus robustus found one amino acid variant in the enamelin protein that varies among the 4 individuals they sampled. The Paranthropus teeth also differ from modern humans in one amino acid, in the col 70 17A1 protein, in which Paranthropus robustus has the ancestral version and modern humans, Neanderthals and Denisovans, have a derived variant. That is the only position that shows Paranthropus robustus to be an outgroup to later Homo in the enamel proteome. In the Homo Naledi sample, the Homo Naledi sample is homogeneous in enamel protein variation. The small number of positions doesn't enable us to say whether this low variation is unusual. Equally small samples of humans sometimes do not vary in more than the AML-Y variant, but some populations do have variation in amino acid sequences that show up in samples of this size. The ancestral Col17A1 variant in Paranthropus robustus is also found in Homo naledi. This confirms earlier work showing that Naledi is likely an outgroup to modern humans, Neanderthals, and Denisovans, because this is an ancestral variant shared with other primates. It does not support a close relationship between Paranthropus robustus and Homo naledi. So far, there is no reporting on this variant in Homo antecessor or Homo erectus data. One interesting possibility for Homo naledi is that the species may have contributed genes to modern humans. The ancestral Col17A1 variant has not yet been seen in the genomes of living people, which at least suggests that this part of the genome did not have a Naledi origin. Okay. Archaeological work underway in the Rising Star Cave System, a photo by John Hawkes. Awesome. I'm taking a sippy sip before I go and parch myself. Okay, not as surprising as it may seem. As you can imagine, how the findings on chromosomal sex of Homo naledi have twisted around my thoughts about the fossil records. We've been studying one of the largest samples of any extinct hominin, and we haven't yet seen a male. Still, it has been known since our first analyses of the Homo naledi sample that the Homo naledi sample is low in variation. In a study led by Heather Garvin, our team estimated body size and stature for adults and suggested that the differences in size between male and female individuals must be very low. None of us were daring enough to suggest that maybe the sample wasn't just a random assortment of the dead. We made the usual assumption, with initially 15 individuals, later close to double that number, we assumed that we were sampling randomly from both sexes. Lucas de Lezen suspected our first assumption was wrong. In 2023, he had led the massive 80-page description of the teeth from the Dinaledi chamber. After spending this kind of time with those teeth, his instinct was that the Homo naledi teeth just do not show as much variation as would be expected in a sample that includes both sexes. Working with several collaborators, including me, he confirmed his intuition with hard data. The me in that point, of course, is Mr. John Hawkes. Okay. Good. The Homo naledi teeth that we found are much less variable than mixed-sex samples of humans, even from single ancient cemeteries. Instead, the variation within a single sex is a better match to the Naledi variation. So the protein data didn't come out of nowhere. The skeletal evidence shows very low variation. The teeth led to the hypothesis that one sex is very strongly overrepresented, and the protein data shows that individuals of female sex are the only ones we have teeth from. Of course, our knowledge is far from complete. Some individuals in the cave system are not represented by teeth in this sample, so we cannot say that every individual definitely was female. However, the neoskeleton from the Lesedi chamber is one of the largest individuals in the sample, and the protein data shows that she was female. So should we change her name, or should we just keep her as Neo? I've seen a couple of experts commenting that males might be hiding in the parts of the sample that have no teeth. To put it simply, there are no bones that are large enough or that manifest other signs of development that would reject a female-only sample. Let's put something here, because otherwise I might get banned or whatever. Henrik, I'm so sorry to hear that you were late, and the shame will indeed haunt your family for many, many generations to come, especially because Denmark was represented twice in this live stream. So, your family will be even more ashamed of you now. As you can see, I put something on yes, okay, so, okay, let's scroll fast, okay. Because, yeah, I don't want my live stream to suddenly, be ruined by YouTube because that was that on a picture in an article that is scientific, but YouTube is like, no, advertisers don't like that. And then your entire stream or video just gets ruined. Awesome. Okay. Reexamining biases. Being involved in this study has changed the way I think about fossil hominins, says Joan Hawkes. I'll probably be commenting at greater length over the next few months. In the case of Homo naledi, the sex bias comes from the behavior of the ancient species, but the analytical biases, the biases and assumptions, those come from researchers. Knowing that the sample is made up of female individuals of all ages gives a great deal of insight about the biology of Homo naledi, which we are following up along several lines of analysis. In previous analyses, we talked about species averages. This might have seemed reasonable, but it was wrong. We don't know the average body size, brain size, or tooth size of Homo naledi. Every comparison of Naledi to other hominin species is affected to some degree by this bias. The Homo naledi sample is far from the only example of statistical bias in the hominin fossil record. I've commented on the male bias of known Denisovan skeletal remains, which are a big impediment to understanding the composition and relationships of this group. Neanderthals are another example. With many more well-preserved adult male individuals, many researchers have discussed this bias in the sample, some concluding that cultural practices, including mortuary treatment, might contribute to the overrepresentation of male individuals. I've had a number of colleagues ask me what the sex estimation of this large sample may mean for the interpretation of mortuary evidence in the Rising Star cave system. I'm a very strong believer of finding multiple lines of evidence and understanding how together they support or reject a hypothesis. In our work on what happened to these Homo naledi individuals, we have relied upon stratigraphic, sedimentological, geological, spatial, skeletal, taphonomic, and demographic evidence, among others. Those analyses come from methods independent of the data on the sex of the individuals. Yet, sex does matter a great deal to understanding the pattern of evidence. It is an analytical bias to assume that culture was not part of the behavior of a close human relative. We evolved from cultural species, and when we find traces of culture, that is not surprising. Culture has shaped the evidence that we find, and our evidence highlights an especially marked case of how skewed evidence may become as a result of ancient cultural practices. Mortuary practices in recent humans, in Neanderthals, and in other archaeological settings do sometimes involve biases in representation or treatment of female and male individuals, or adults and children. One of the largest early death assemblages of hominins is from Cima de los Huesos in Spain, which has a very strong bias against the presence of children. In the Rising Star situation, we have the expected number of children, around half the sample, and a very strong bias toward female individuals of all ages. Cultures vary enormously. Sometimes the interactions of mortuary and sex reflect much broader patterns of treatment or beliefs. Sometimes they don't. Sometimes the intersection of mortuary with the sex of the deceased is counterintuitive, where the sex with lower status has higher investment after death. Whatever stereotypes people might have about mortuary practices, they couldn't match the reality of how diverse those practices are. No humans are Homo naledi. We don't know that the variation in humans makes much sense of a model for naledi cultural practices, whether looking at living groups or historical ones. But as we find more evidence, what I am finding says John Hawkes. What I'm finding the most engaging is how what seems unusual, one headline said baffling, is mostly because it reveals how our assumptions were wrong. Assuming that every sample is a random assortment of individuals is a bias. Assuming that the typical or most complete skeletons are males is a bias. Assuming that the typical or most complete skeletons, assuming that cultural phenomena did not apply in past societies is a bias. It's a challenge to re-examine your biases, but it's essential to scientific understanding. And again, I love a good John Hawkes article because he writes in a very, very, very compelling way. It reads it's very fluid, and I like that a lot. So, yeah. That was it. That was what I wanted to cover today. So yeah, if you have not seen my previous video, because apparently almost no one did, it is my worst performing upload in almost 5 years. It's this, the paleo fantasy caveman myth that just won't die. It doesn't even have 10,000 views in the first almost 5 days. I want to cry into a corner. I have to admit, I uploaded that on Sunday, 8 p.m. my time, and I immediately realized that this video was going to completely flop, performance-wise, because the video itself, I'm very happy about the video. The video is great, like the quality is great, the subject is awesome. YouTube has not recommended it to almost all of my viewers. Most of the comments I received were people saying that they weren't recommended it, they only saw it because they went to my channel. And I have to admit, I saw on Sunday evening that the video was going to flop, and Monday morning I woke up and I saw that the video was actually completely dead and not gaining any views, and I cried. And I cried honestly for a couple hours because I was really hurt. I spent 3 weeks researching for that video. I have never had a sources list that long. Like 3/4 of my description is just the list of sources that I used for that video. And then there are, of course, a lot of men that only focus on the cartoon exaggeration of the paleo fantasy trope, the courtship by club. So yeah, I don't know what went wrong. I don't know if this was like an actual YouTube problem, issue, whatever. But I just know that I cried really, really hard when it came to that video. And yeah. A lot of people tell me that they were not recommended that video. So please, please, please do me a favor. If I can ask you guys for a favor that's free and only takes about half a minute of your time, watch that video and share it wherever you can. Share it with your friends, your family, people who are interested in anything. Share it on a public forum. Share it on Reddit or Threads or Blue Sky or Twitter or whatever you use. But please, please, I beg you, share it. The video is so good, and the underlying message of that video is so strong, and it is such a societal issue that we still have. And I understand that me attacking a certain societal issue isn't very welcoming in certain parts of the world at the moment, but please, please share it. And if you haven't seen the video previous to that, the Dolmen of Guadalperal, please go watch that too, because that used to be my worst upload in almost 5 years, and then this one went like, oh hey, I can be even worse than that. So yeah, okay. I don't know. Okay, so that said, again, check out the merch store, historywithkaylee.com. Check out consider joining me in Turkey. I promise you it's going to be amazing. It's a small group. I usually go with small groups to Turkey. We're not with like 25 people and you don't have time to even talk with me. I sit with you from breakfast until dinner. And like usually after dinner, I hang around for a little bit before I go to my room, unwind and recharge for the next day. But like, please consider joining. Because yeah, I like to travel with you guys. I really enjoy showing Göbekli Tepe, Karahan Tepe, Sayburç, Sefer Tepe, Harran, the Bazda Caves, Şuayb, Sokmatar, Mount Nemrut, a boat tour on the Euphrates, the fairy chimneys at Cappadocia, the underground city of Kaymakli, and of course the Hittite capital of Hattusha. I enjoy showing all of that to you, and we usually go to one or two sneaky places that we don't tell prior to the tour. That's like a cherry on top when you join, and we give you a little sprinkle of extra. The first year that was Sefer Tepe. We now added that standard. Last year that was Harbetsuvan Tepe, not open to the public at all. And we just went there and decided maybe the archaeologists will let us in. And the head archaeologist did let us in. So like we try, you know? We try. And yes, it is an annual tour. So if you cannot make it this year, I will start promoting the tour for next year around January. So yeah, keep ahead. Keep close eyes on that. And yeah, I think with that said, I am going to sit in a lukewarm shower, because cold showers while it's hot is really not good for your body, because the temperature difference is too big. So like you're like shocking your body to cool down, even though cold skin keeps the heat inside of your body. Not good. No. Lukewarm or warm showers, like a little bit mild, that's the best, because it still opens up your pores, it cools you down a little bit, and you can still release the heat through your skin. Tip from me. I've been dealing with these weird hot summers in the Netherlands for a while now. If you do the cold shower method, why would you shock your body like that? Also, do not give your animals ice cubes directly. And if you do maybe 1 or 2 ice cubes in a large bowl. Usually cats and dogs don't really actually like the ice cold water. Again, it's a shock to the system. It's not good for them. It's not good for you. You don't want that. Fresh water is the only thing they need. Doesn't necessarily have to be cold. So yeah. And if you have cats, especially a little bit of older cats in this heat wave, and you give them wet food regularly, put a little bit of water with the wet food, like mix it in. Trust me, that's what my vet always said to me. Like, do a little bit. If you enjoy the shock, El Guapo, then who am I to burst your bubble? Enjoy the shock. But yeah. Cooling your wrists, also one of the best ways to cool down, because your veins are located very close to the skin. It helps cool your blood from the inside, so cool your wrists, the inside. That helps. If you have like these sports bandages or bracelets things, you can make them cold and wet and put them on your wrist, and it helps cool you down as well. So yeah. Okay, those were my keep cool tips. Open your doors and windows opposite from each other on different levels of the house after the sun goes down, which is around 10:20. 10:15, by the way. It's 10:15 p.m. that the sun goes down in the Netherlands at the moment, and it rises around 5:10 in the morning. So yeah, a little bit more than 6 hours of no sunlight that we have. So yeah, besides that, we're just heating up and melting away. Stay hydrated, drink, and I will see you in the video premiere, probably either on Saturday or Monday, because I have decided to stop uploading on Sundays. So you will not have a Sunday video from me for the foreseeable future. I will not upload on Sundays, because my last 2 uploads on Sunday, as you can see, completely flopped. And the 2 videos before that were uploaded in the middle of the week on a Tuesday and a Wednesday, and they did great. And the video before that, that also flopped, was also uploaded on a Sunday. So I stopped uploading on Sundays. Sorry. So I'll upload on any other day of the week whenever I feel like it, but no longer on Sundays. Yes, unfortunately, this is the end of the stream. But you can watch the stream back and hear all about this major news about Homo Naledi and the fact that we might have never found a Homo Naledi male skeleton. What? That's really cool. Okay, so guys, girls, gents, ladies, lads and laddies, love you all, and I will see you in the video premiere in a couple days, and fingers crossed for the Netherlands tonight, because we play Tunisia. I mean, we should win. All things considered, I think we should win. But it depends, do we get to the first place in the group or the second place in the group? And that depends on Japan and how much they will score against Sweden. Or if Sweden wins from Japan. Could be cool too. Yeah. Okay, so I'll be awake for a couple hours because the game in Kansas also might be delayed because there's a storm coming there. So yeah, okay, cool. Okay, cool. I'll see you all soon, and I hope you have a great day, great Thursday. If you're in the Europe, keep cool, be okay. If you're in the US, stop telling people in Europe to buy AC. Doesn't work like that. Okay, love y'all. Bye.